Hi, Bhaskar this side. Today we are going to talk about what is the relationship between the pKa of drug substance and its absorption. Isn't it important to understand in case if you are having a suitable dissolution but still you are not able to meet the bioequivalence requirement? So let us understand what is the role of pKa of drug substance to its absorption through the GI tract. So when it comes to gastrointestinal tract, the pH of gastrointestinal tract is from let us say 1 to 7.5, and between this pH range, the small intestine is considered to be a major reason for the drug absorption, but that doesn't indicate that only small intestine needs to be targeted. The absorption process is sometimes dependent onto the dissolution, but once the drug substance goes into a solution form, then a dissolution do not remain the rate limiting step. The nature of your drug substance, whether it is ionic or non-ionic, is actually going to impact drug absorption. So stronger absorption is possible in case if your drug substance is into non-ionic state, and the weaker absorption is possible in case of your drug substance is into ionic state. See, let us understand there is a semi-permeable membrane between the GI fluid and to the bloodstream. And this semi-permeable membrane is the lipophilic. So what kind of drug substance is or what kind of compound can transfer easily from GI fluid to bloodstream through so-called lipophilic membrane? Of course, the drug substance or compounds with lipophilic nature or the compound with non-polar nature like the non-ionic compounds, because non-ionic compounds are non-polar in the nature, whereas the ionic compounds are polar in the nature. So the ionic compound will prefer to stay in the GI gastrointestinal fluid, whereas the non-ionic compounds will prefer to transfer through the semi-permeable membrane and enter into the bloodstream or the systemic circulation. That is the reason why the non-ionic compound has the stronger absorption, whereas the ionic compound has the weaker absorption. So we understand one thing very clearly, that the moment the drug comes into a solution form after dissolution, and as we said that the dissolution is not going to be a rate limiting step for the absorption, but the further barrier can be this ionic state of the compound. Now, what is the reason that your compound is into ionic form or your compound is into a non-ionic form? See, you cannot control that. It can be controlled by the pH and the pKa. So what is the relationship between the pH and the pKa? Acidic compounds are generally found to be non-ionic into the acidic pH, whereas basic compounds are found to be non-ionic into the basic pH. So acidic compounds will be into ionic state in the basic pH, whereas the basic compounds will be into ionic state into the acidic pH. Now this is very broader aspects of understanding the ionization state of weakly acidic or weakly basic compound. Now let us understand with the specific value of the pKa and the nature of the compound, whether it's acidic or basic. Now what is going to be the ionic state, and based on to that we will able to understand its probability of absorption. The first example here is the stronger acid with the pKa less than 2.5. So what is going to be the state of ionization? Now this compound will get completely ionized across the GI tract pH. And if it is completely ionized, it is already a less preferred to get transferred through this semi-permeable membrane, because the semi-permeable membrane is the lipophilic membrane. But the moment your drug substance is into ionic form, it is actually not the lipophilic but the hydrophilic. And hence it is going to prefer the hydrophilic fluid of the GI tract rather than getting transferred into the systemic circulation. And hence, if your compound's pKa is less than 2.5 having stronger acid, it is going to be a poorly absorbed from entire GI tract. The next point is, in case if a compound is very weak acid having pKa greater than 8. Now as we know that these weakly acidic compounds can get ionized into the alkaline pH, but the pH must be at least 2 unit greater than the pKa value. Now here we said that the pKa of the compound is greater than 8, or let us assume if it is 8, this compound can get ionized into the pH of 8 plus 2 becomes 10. But does the 10 pH really exist into the GI tract? It is not. The pH range is from 1 to 7.5, and hence this compound will be unionized at all pH values. It will be a non-ionic compounds across the GI-pH tract, and hence it can get very easily absorbed along the entire length of the GI tract. The next example is moderately weak acidic compound with the pK value of around 2.5 to 7.5. Now being acidic compound, this compound will be unionized into a gastric fluid, but it can have the partial ionization into the intestinal fluid. Hence, this compound can be better absorbed from the gastric fluid or the stomach. The next important point is the stronger base when the pKa is greater than 11. So these compounds will have the ionization across all the pH ranges of the GI tract fluid, and hence it will be poorly absorbed from the GI tract fluid. The last one is the moderately weak base with the pKa value of between 5 to 11. So this compound will be ionized at gastric pH. Relatively, it will be unionized at the intestinal pH. So this compound will get better absorbed from the intestinal fluid. So once you understand the site of absorption, you can also think of selecting the suitable dissolution medium where it can reflect the pH condition of the site of absorption. For example, in case if the compound is getting absorbed along the intestinal fluid, so you can think of the pH of the intestinal fluid as a dissolution medium pH. In case if your compound is getting absorbed from the gastric fluid, then the gastric fluid pH can become your preferred dissolution medium's pH. But it is not a mandatory to have this correlation during the dissolution method development, but this is just an advice to have the dissolution condition in the place. I hope you must have now understood what is the relationship between the drug pK value and its absorption. Thank you very much for watching this video, and I will meet you soon with such kind of another very useful and informative videos. Till then, take care and bye bye. See you soon.